Okay, so our next talk is Structuring Genetic Disease Complexity and Environmental Drivers by Lynn Schriml.
I think the order of the talk was quite nicely set up. So the last talk was talking about the need to look at the genetic and environmental underpinnings of disease, and that's very much what we're talking about today. So the disease ontology was started 15 years ago. It's our hometown, Chicago, at Northwestern University, so it's quite nice to be back. Today I'm going to highlight what we've been doing in the last year and next 4 years as a driver of our project. So in particular, the disease ontology, we've always had a very structured, is a hierarchy, that's what you're seeing in the middle of the screen, to look at the classification of disease based on etiology. Now what we've been diversifying into doing now, under U41 from NHGRI, is to look at the anatomical location of diseases, the genetic underpinnings, and environmental drivers. And so if you look at the ontology lookup service, which is a picture on the right, you can get different views of the ontology to see how diseases are related based on these elements of the information. So we're continuing to move forward with this, and today I'm trying to outline how we're using the various biomedical ontologies to strengthen these identifications of data. It's a clinician team-led approach, and we're using a stepwise approach, and I'll describe that and what we're moving forward. And oh, by the way, I do have a poster later, and I have stickers if anyone wants any disease ontology stickers. So I'll be there at 6 o'clock. Okay.
What's the number
of
your poster?
Oh, sorry, 575. Well, I'm right next door. So just to give you an idea, so this is very much a stepwise approach as we laid it out. So to figure out the scope of what we would do and change, because we're really expanding the content and the ideas of the ontology quite a bit. So we have a 5-person clinician team that's leading this aspect of it. At the top of the screen, you can see one of our objectives is to figure out how do we capture this data model. From a clinician's point of view, we want to do differential diagnosis, and they look at an ontology. How does it make sense then to look at genetic disease? So we've gone over this for several months, and this is the basic data model that we've then come up with. Going from a data model to an ontology, it's a big step. The in-between is looking at a number of key complex genetic diseases and how would you encode those. These are down to the right. The ones that are in bold are the ones we've already gone through. We meet monthly with a clinician team. They research a particular disease. We discuss it for a couple of hours. It's quite fun, actually. And figure out how are the different aspects of disease seen in clinic. And I'll show you one example with Prada Willi disease syndrome. So on the bottom slide, the slide over to the left, we go from this complex disease model. We restructure, look at how we restructure this disease knowledge. We use these driving use cases, and then we're building up a framework for differential diagnosis. That's the whole intent of it. So we're using driving biological, not projects, but diseases in this case, to really make sure our data model makes sense for the biology and the clinician, and then we make sense for it in the ontology. That's another part of this talk, the next slide. So a big step for this is what other biomedical ontologies do we bring in that we encode to capture this data? Working with the immune epitope database, this last year we were doing this as well. We're looking at foods, chemicals that are needed to define allergic triggers. So part of this work is defining new relations. So in the middle of the slide, you're seeing various relations we're using in this aspect of it. But for the work we do with IEDB, we had to identify a relation has allergic trigger, and that's part of the ontology. This allows us to build up these axioms to have more refined specification of what the aspects of the disease are. On the left, I'm showing you the ontologies we're using for environmental drivers, the others for complex disease, and the ones we're continuing to add in. And we have one more relation we'll be proposing and developing this year is the environmental trigger one. So as I was saying, it's a staged work. Most of the work in this last year has been on complex diseases, and we're getting into the environmental work now. So to look at one example for Prader-Willi syndrome, it's a very complicated disease. Yes, there is chromosomal changes that occur, but when a patient comes into clinic, they can have one of a variety of the genetic backgrounds that cause the disease. And so we really want to capture that. You can even have cases that are fully syndromic, where you have a set of symptoms and phenotypes, but there's no genetic change that you can detect at that time. So we want to really capture it. And we're also looking at what are the predominant genetic backgrounds. So in this case, a particular paternal deletion on 15q is 70% of the cases. We want to get the others as well. And so that's what you're seeing on the next slide, and we're encoding in the ontology itself. So to do this in an ontology, you also have to build up these logical definitions, these axioms from the top down, because it is a hierarchical structure. So we start with disease and work our way down. How do we redefine genetic disease in this basis? We use a new relation called disease has basis in, and this is from the relations ontology. And we figure out what is the most appropriate description to have for that. On the ontology, it's a whole separate discussion in one of our working groups. On the ontology side, how do you capture this rigorously? How does it make sense biologically? And these are the ones we've integrated into the system to be able to do something like this. So you can have a situation where all of these descriptions are an and situation, which means the patient, every instance of this disease, and every patient would have every one of these. In Prada, that's not the case. We do the or, which is at the bottom. Each individual will have one of these. So the reason we're kind of working this system out and figuring out how to actually encode it on the ontology side is you're going to have patients in certain diseases where they will have all of the above, or all of the symptoms, or you have cases where they have this set of symptoms of 3 and 2 phenotypes over there. So we set up this model so we can encode all that, not just for genetic diseases, but for symptoms, for phenotypes as well, and the other elements that we're capturing. So it's quite fun to do this development on that side, but it's all being engineered and driven by the clinical side.
1 minute.
Perfect. And just one mention, we are working on the exposures for environmental diseases, and so far we are working on collecting the data, figuring out the data model, and then working from different data sources to really establish what are going to be the drivers and how we'll encode them. But we've done the ontology work already to kind of get that aspect of it. And just on the last slide, it's just as we're leaving, we're a community-based organization. For 15 years, we work with groups that have data needs. Much of what you're seeing today was driven by people that have requests, working with different community groups so that we actually encode the data that's needed to be encoded in the right way. So please, if you have suggestions, come see me, chat with me, talk to us at our system where we have lots of ways of contacting us people are familiar with. And if you're not, I'll be at the posters later. So thank you very much, and thank you for using the DOE.
We have time for one question, if there is one. Oh.
So it's great work. I'll definitely come by the poster and ask more questions. I'm a little concerned about the anatomy. Mm-hmm. So a disease is a process, anatomy is a thing, and lots of diseases affect lots of anatomy. So
Right.
how are you going to tackle that? It's not just is located in.
So we will talk about where a disease is initially located in the body. Like you said, many diseases are located in many locations. But when you're thinking of it from the clinical point of view, where is it located in the body when the person is first coming in and getting diagnosed? We don't look about, because we are looking at these as classes of diseases, it's where the disease occurs first. So metastases and things like that, we're not encoding within it, or where diseases move to. We use the Uber on ontology for our anatomy.
Great. Thank you very much, Lynn. Okay.